welcome to today's session on microbiological testing. We'll be studying another chapter from the microbiology manual, that is, the detection and confirmation of Salmonella species in food and water samples. This process is important for ensuring food safety as Salmonella is common cause of foodborne illness. So let's get started. First, let's go over the equipments and the media which will be required to carry out this procedure. You will need basic microbiological tools like petri dishes, pipettes, and incubation oven. Along with that, you will require different kinds of culture media such as lactose broth, trypticase soya broth, trypticase soya broth containing potassium sulfite at a final concentration of 0.5%, reconstituted non-fat dry milk, 1% aqueous brilliant green dye solution, selenite cystine broth, tetrathionate broth, xylose lysine deoxycholate agar, hectation enteric agar, bismuth sulfite agar, TSI that is triple sugar iron agar, lysine iron agar, urea broth, phenol red dulcitol broth, phenol red, lactose broth, tryptone broth, casein broth, malonate broth, buffered glucose for BP and MR test, brain heart infusion broth, and buffered peptone water. Next, we will prepare the food sample. We have to take 25-gram sample and aseptically transfer it into sterile container which will be containing 225 ml of sterile lactose broth. Buffered peptone water, trypticase soya broth and nutrient broth can also be used for pre-enrichment process. If we are analyzing water sample, then we have to take 250 ml of water sample for the same process. After that, if we are considering solid food sample, we have to make a uniform suspension by blending if necessary. The container which we have taken should be of a wide mouth and it should have a screw cap. After blending, the cap container is capped and it is let to stand at room temperature for 60 minutes. Instead of lactose broth, there are various pre-enrichment broths which are being used for various different kinds of food samples. For example, if we want to analyze Salmonella species in non-fat dry milk and dry whole milk, then we can simply use sterile distilled water with 0.45 ml of 1% aqueous brilliant green dye, which should be added into that before incubation. If we want to analyze for dried active yeast, then we can use trypticase soya broth. If we are analyzing onion garlic powder, then we can use trypticase soya broth containing potassium sulfite at a final concentration of 0.5%. If we are analyzing Salmonella species in milk chocolate, then should be reconstituted using non-fat dry milk. After doing all these processes, we have to shake the samples and adjust the pH if necessary between 6.6 to 7.0, with sterile 1 normal NaOH or 1 normal HCl. After which we will incubate the pre-enriched samples at 35 degrees Celsius for a period of 24 hours. Next, we have to carry out selective enrichment to select the Salmonella species from other food contaminants which might be present in our food sample. For that, we have to transfer 1 ml of incubated sample into selenite cysteine broth and another 1 ml into tetrathionate broth. This media will inhibit the growth of other competing flora which might be present in our food sample while promoting the growth of Salmonella species. The tubes are then incubated for another 24 hours at 35 degrees Celsius. After which we have to do selective media plating. So, in this step, we will be using selective media such as your XLD, HEA, and BSA agars to isolate Salmonella species which might be present in your food sample or water sample. What we need to do is we have to streak a loopful of the incubated broth on the selective agar plates such as XLD, HEA and BSA. After that, we have to incubate these plates at 35 degrees Celsius for a period of 24 to 48 hours. And then we will examine the plates for the characteristic Salmonella colonies. On XLD agar, Salmonella appears as pink colonies with black centers. On HEA, the colonies are blue-green with